In the quiet darkness between stars, something remarkable persists. Not urgency, not drama, just the patient, unhurried possibility of existence beyond our small world. Tonight, we'll explore what science has quietly discovered about life in the universe. Not through speculation or imagination, but through careful measurement, thoughtful analysis, and the simple recognition that the cosmos operates according to patterns we're only beginning to understand. This is a journey through verified facts, through numbers so vast they inspire calm rather than excitement, through discoveries that suggest we may not be as alone as the night sky sometimes makes us feel. There's no rush here, no deadlines, just the slow unfolding of cosmic truth, the gentle accumulation of evidence, and the recognition that the universe has been conducting its experiments with life for far longer than our species has existed. So settle in, let your breathing slow. What follows is not a story of aliens arriving, or signals received, but something perhaps more profound. The realization that the ingredients, the spaces, the conditions for life appear again and again throughout creation, waiting patiently in forms we're learning to recognize and measure. The universe, it turns out, is not hostile to life. It's simply vast beyond imagining, old beyond measure, and operating on timescales that make human urgency look like the frantic buzzing of a mayfly at noon. Let's explore what that means. When we look up at the night sky on a clear evening, we see perhaps a few thousand stars with our unaided eyes. It's beautiful, it's humbling, and it represents essentially nothing compared to what actually exists beyond our small sphere of perception. Our galaxy, the Milky Way, contains somewhere between 200 billion and 400 billion stars. That's not a round number chosen for convenience. It's an estimate based on careful measurements of galactic mass, luminosity, and structure. Estimates suggest between 200 billion and 400 billion stars populate our galaxy alone. Each of these stars represents a sun, many potentially surrounded by their own family of worlds. But that's just our galaxy, one galaxy among countless others. Current observations suggest the observable universe, the portion of existence from which light has had time to reach us, contains somewhere between 100 billion and 2 trillion galaxies. The observable universe contains as many as an estimated 2 trillion galaxies. The uncertainty in these numbers isn't sloppiness. It reflects the genuine challenge of counting objects so distant that many appear as faint smudges even to our best instruments. Let's sit with that number for a moment. 2 trillion galaxies, each containing, on average, hundreds of billions of stars. When astronomers attempt to calculate the total number of stars in the observable universe, they arrive at figures around 10 to the 24th power. The observable universe contains as many as an estimated 10 to the 24th power stars. That's a 1 followed by 24 zeros. More stars than all the grains of sand on every beach on Earth. More stars than seconds have passed since the universe began. This isn't meant to make you feel small. Rather, it's meant to convey something else entirely, that with such abundance, the question isn't whether the conditions for life exist elsewhere, but rather how many times they've been replicated across that incomprehensible expanse. The Milky Way spans over 100,000 light-years in diameter. Our Milky Way galaxy spans over 100,000 light-years in diameter and contains approximately 400 billion stars. Light itself, traveling at 186,000 miles per second, would take 100,000 years to cross from one edge to the other. The distances between galaxies are even more staggering, measured in millions or billions of light-years. In such a cosmos, repetition becomes not just possible, but inevitable. If the basic ingredients of life exist, and we know they do, then given enough space, enough time and enough variety in conditions, those ingredients will combine and recombine in every possible configuration. Some configurations will fail. Others will persist. A few might flourish. Recent discoveries have only reinforced this sense of cosmic abundance. The Hubble Space Telescope, peering at what seem to be empty patches of sky, has found thousands upon thousands of galaxies in regions we thought were blank. Astronomers came to the surprising conclusion that there are at least 10 times more galaxies in the observable universe than previously thought. Each new generation of instruments reveals more structure, more complexity, more worlds than the previous generation could detect. The James Webb Space Telescope, launched recently, continues this tradition of discovering abundance. Its deeper vision into infrared wavelengths shows galaxies forming in the early universe, barely a few 100 million years after the Big Bang itself. When we observe the universe at great distances, we see it as it was in the distant past, smaller, denser, hotter, and less evolved. These observations tell us that galaxy and star formation began early and has continued persistently for over 13 billion years. What does this mean for life? It means that if the probability of life emerging on any given suitable world is even 1 in a trillion, there could still be trillions of instances of life scattered throughout existence. The numbers work in favor of life not because life is common, necessarily, but because the cosmos is so extraordinarily, almost incomprehensibly vast. Consider the nearest large galaxy to our own, Andromeda, visible as a faint smudge to the naked eye on dark nights. It lies approximately 2.5 million light-years away. The Milky Way is separated from the Andromeda Galaxy, its nearest large neighbor, by just over 750,000 parsecs, or 2.5 million light-years. That galaxy contains roughly 1 trillion stars. Between Andromeda and the Milky Way, and within our local group of galaxies, hundreds of smaller dwarf galaxies orbit and interact. Each represents another vast laboratory where the chemistry of existence plays out. Even within our own galaxy, the stars are distributed across an enormous volume. Most stars are separated from their neighbors by distances of several light-years. Our nearest stellar neighbor, Proxima Centauri, lies just over 4 light-years away. A distance light takes 4 years to traverse, or over 25 trillion miles. About 40 planets, including the nearest extrasolar planet, Proxima Centauri b, have been found that are both roughly Earth-sized and orbiting within the habitable zones of their stars. That star hosts at least 1 planet, and that planet orbits in the habitable zone where liquid water could exist on its surface. These distances mean that even if intelligent civilizations exist in our galaxy, they might be separated by spans so vast that communication would take decades or centuries. A signal sent from Earth to Proxima Centauri takes 4 years to arrive. A response takes another 4 years to return, and Proxima Centauri is our closest neighbor. Most stars in our galaxy are thousands or tens of thousands of light-years distant, but this immense separation doesn't argue against life. It simply means that life might exist in quiet isolation, evolving independently on countless worlds, each developing its own solutions to the challenges of existence. The universe provides not just one laboratory for life, but trillions upon trillions of them, each running its own experiment with slightly different initial conditions. The scale of the universe also provides something else: time. Our Sun is approximately 4.6 billion years old. Every star has a habitable zone, but where that zone lies is different for stars of different sizes and brightness. But many stars in our galaxy are significantly older. Some approach 13 billion years, formed when the universe itself was less than 1 billion years old. If life emerged on a world orbiting one of those ancient stars, it would have had billions of years more than Earth's life to evolve, adapt, and develop. The cosmic census continues to grow more detailed as our instruments improve. Projects like the Gaia mission have precisely measured the positions, distances, and motions of over 2 billion stars in the Milky Way. The ESA Gaia spacecraft has observed 2,000 million stars in our Milky Way, monitoring each of its 2 billion target stars 140 times during its mission. This unprecedented mapping reveals the structure and dynamics of our galactic neighborhood in exquisite detail. What emerges from all these numbers is not just scale, but structure. Galaxies aren't random scatterings of stars. They have spiral arms, galactic bars, central bulges, regions where star formation proceeds more actively, where heavy elements are more abundant, where conditions might be more or less favorable for planetary systems. Some regions of galaxies receive more radiation from cosmic sources. Others are quieter, more stable. This variation creates a spectrum of environments, each with its own characteristics. The vastness doesn't make life certain on any individual world, but it does suggest that the right combinations of conditions, whatever those conditions turn out to be, must occur somewhere, simply because there are so many somewheres to try. Nature has run countless experiments across billions of years and trillions of locations. In that context, expecting Earth to be the only success seems, if anything, statistically improbable. This isn't about faith or hope. It's simply about scale, about the recognition that the universe operates through repetition and variation across spans of space and time that dwarf human experience. And in such a universe, the ingredients and conditions for life appear again and again, waiting patiently for the right combinations to occur. The calm we can take from this is profound. We don't need to rush. We don't need to solve the question of alien life tomorrow. The cosmos has been running these experiments for over 13 billion years, and it will continue for trillions more. Whatever the truth about life's distribution, the universe has time to reveal it slowly, carefully, through patient observation and thoughtful measurement. Life as we know it on Earth is built from a relatively small set of elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. These 6 elements form the backbone of every living thing on our planet, from bacteria to blue whales. They combine into amino acids, which link together into proteins. They form the nucleobases that make up DNA and RNA. They create the lipids that form cell membranes. For life to exist elsewhere, similar building blocks would need to be available. Not necessarily the exact same molecules. Evolution might find alternative solutions on other worlds, but at least the raw materials from which complex chemistry can emerge. The question then is simple. Are these materials rare in the universe or common? The answer, it turns out, is that they're remarkably common. Space is not empty. Between the stars, throughout interstellar clouds, within the disks of gas and dust around young stars, and embedded within meteorites and comets, organic molecules, molecules containing carbon bonded to hydrogen, exist in tremendous variety and abundance. Samples returned from the asteroid Bennu by the OSIRIS-REx mission detected amino acids including 14 of the 20 used in terrestrial biology, amines, formaldehyde, carboxylic acids, polycyclic aromatic hydrocarbons, and nitrogen heterocycles, including all 5 nucleobases found in DNA and RNA. This single sample from one asteroid contained not just 1 or 2 simple organic molecules, but a diverse suite of complex compounds, the very building blocks that life on Earth uses to construct itself. Think about what that means. An asteroid, a cold, lifeless rock drifting through space, naturally contains molecules that on Earth are associated exclusively with biology. The molecules themselves aren't alive, of course, but their presence demonstrates that the chemistry leading toward life isn't unique to Earth. It happens naturally in space, without any need for living processes to guide it. The Bennu samples revealed something else equally important. Samples from asteroid Bennu are volatile-rich, with more carbon, nitrogen, and ammonia than samples from asteroid Ryugu and most meteorites. These volatile-rich compositions suggest that such materials are not rare oddities, but represent a common class of small bodies in our solar system and, presumably, in other solar systems as well. Comets, too, carry organic materials. The European Space Agency's Rosetta mission, which rendezvoused with Comet 67P Churyumov-Gerasimenko in 2014, made a historic discovery. Rosetta made direct, repeated detections of glycine in the fuzzy atmosphere or coma of its comet, marking the first unambiguous detection of glycine at a comet. Glycine is the simplest amino acid, a fundamental component of proteins. Finding it in a comet demonstrated that amino acids can form and survive in the cold, radiation-bathed environment of deep space. The Rosetta mission found more than just glycine. The multitude of organic molecules already identified by Rosetta, joined by the exciting confirmation of fundamental ingredients like glycine and phosphorus, confirms the idea that comets have the potential to deliver key molecules for prebiotic chemistry. Phosphorus, essential for DNA, RNA, and the energy-carrying molecule ATP, was detected, alongside the amino acids. These weren't trace contaminants. They were present in measurable quantities, formed through natural processes in the cold depths of the solar system. Meteorites that fall to Earth tell a similar story. The Murchison meteorite, which fell in Australia in 1969, has been intensively studied for decades. Analysis revealed over 90 different amino acids in its matrix. The variety and complexity of meteoritic organics is becoming clearer as more meteorites are studied. Only a fraction of these amino acids are used by life on Earth. The rest are chemically similar but not identical, demonstrating that the processes creating them are not biological but arise naturally from chemical reactions in space. What's particularly striking is the diversity of these molecules. The Ryugu asteroid samples, returned by Japan's Hayabusa 2 mission, contain dozens of different organic compounds. All life on Earth contains amino acids and carbonaceous chondrite. Meteorites have been suggested as their source at the origin of life on Earth. The samples showed that amino acids can form through aqueous alteration, chemical reactions involving water, occurring within asteroids themselves. These parent bodies essentially become tiny chemical laboratories, mixing and recombining elements over millions of years. The process isn't mysterious. When planetesimals, the building blocks of planets, formed in the early solar system, they incorporated water ice and simple organic molecules from the interstellar cloud that gave birth to the Sun. As these bodies collided and merged, the heat and pressure generated created conditions for more complex chemistry. Upon formation of planetesimals in the protoplanetary disk, amino acid-bearing ices could have been accreted into such bodies along with other organic compounds and inorganic materials. The ingredients were baked in from the beginning. One particularly important molecule detected in space deserves special mention. Scientists from Japan and NASA have confirmed the presence in meteorites of a key organic molecule which may have been used to build other organic molecules, including some used by life. This molecule, hexamethylNTretramine, or HMT, is stable even in the harsh conditions of space. It can break down in the presence of liquid water to release formaldehyde and ammonia, two molecules that can then react to form amino acids through well-understood chemical pathways. The presence of HMT solves a puzzle that had troubled scientists. Formaldehyde and ammonia are highly volatile. They evaporate easily. How could they persist long enough in asteroids to participate in the chemistry leading to amino acids? The answer: they don't need to persist. They're stored safely in the form of HMT and released only when conditions are right. This kind of chemistry, protective molecules storing reactive components until needed, demonstrates a level of sophistication in natural space. Chemistry that surprises even experienced researchers. The universe appears to have its own methods for preserving and delivering the materials needed for life. The sheer abundance of organic material in space is staggering. Observations of interstellar dust clouds have revealed complex organic molecules, polycyclic aromatic hydrocarbons, formaldehyde, methanol, and many others, drifting between the stars. These molecules form through photochemistry, driven by ultraviolet light from nearby stars acting on simpler compounds. Over time, more complex molecules build up, coating dust grains and eventually becoming incorporated into new generations of stars and planets. When new solar systems form, these organic rich materials are present from the beginning. The disk of gas and dust swirling around a young star contains all the elements and compounds needed for life. As planets coalesce from this disk, they naturally incorporate these materials. Earth certainly did. The early bombardment that our planet experienced brought not just water, but also organic molecules by the ton. What makes this particularly relevant to the search for alien life is the recognition that this process isn't unique to our solar system. Every star forms from a similar disk. Every planetary system incorporates similar materials. The basic ingredients for carbon-based chemistry are delivered as standard equipment, a natural consequence of how stellar systems form. Recent sample return missions have provided unprecedented detail about these materials. The OSIRIS-REx mission to Bennu and the Hayabusa 2 mission to Ryugu both returned pristine samples untouched by Earth's atmosphere or biosphere. Missions such as NASA's OSIRIS-REx and JAXA's Hayabusa 2 will provide samples from primitive bodies with greater contextual information than typically studied meteorites. These samples allow researchers to study prebiotic chemistry with confidence that they're observing extraterrestrial materials, not contamination. The results confirm what spectroscopic observations had suggested: organic chemistry happens naturally and abundantly in space. The molecules required for life don't need to be manufactured through biology. They arise spontaneously through straightforward chemical reactions when the right elements are present in the right conditions. Phosphorus, that essential element for DNA and energy metabolism, has also been detected in space. Phosphorus, a key component of DNA and cell membranes, was detected at the comet alongside amino acids. While less abundant than carbon or nitrogen, phosphorus exists in sufficient quantities in asteroids and comets to supply planetary surfaces with this crucial element. Even more intriguing are recent discoveries of complex organic molecules in the plumes ejected from Saturn's moon Enceladus. Scientists found new complex organic molecules spewing from Saturn's moon Enceladus, a clear sign that complex chemical reactions are taking place within its underground ocean. The newly detected molecular fragments included aliphatic compounds, nitrogen-bearing compounds, and oxygen-bearing compounds, the same types of molecules that on Earth participate in the chains of reactions leading to biological molecules. Enceladus isn't unique. Europa, Jupiter's ice-covered moon, almost certainly has a similar subsurface ocean. Titan, Saturn's largest moon, has lakes and seas of liquid methane and ethane on its surface. Each of these bodies represents a different environment where organic chemistry can occur. Each might be exploring different pathways toward complexity. The point isn't that any of these places necessarily harbor life. The point is that the raw materials, the organic molecules, the water, the energy sources, are present throughout our solar system and by extension throughout solar systems everywhere. The chemistry of life isn't exotic, it's ordinary, woven into the fabric of cosmic evolution. This realization brings a certain peace. We're not searching for something impossibly rare. We're looking for variations on chemistry that happens naturally throughout the universe. The ingredients are there, the conditions exist, the processes run continuously. Whether those processes culminate in life depends on details we're still working to understand, but the foundation, the basic building blocks, is universal. As we continue to study meteorites, comets, and asteroids, the catalog of organic molecules only grows longer. Each new mission reveals more complexity, more diversity, more evidence that the chemistry leading toward life is not a rare miracle, but a common feature of cosmic evolution. The molecules float between stars, settle onto planets, react and recombine in endless variations. Some reactions lead nowhere, others persist. A few might cross the threshold into self-replication, adaptation, and ultimately life. The universe, it seems, has been running experiments in organic chemistry for over 13 billion years. Every star system represents a new trial, a new combination of conditions and materials. The results of those trillions of experiments are still being tallied, but the sheer abundance of starting materials suggests that interesting chemistry, perhaps even biological chemistry, must occur more often than we once imagined. For most of human history, we assumed life required narrow, gentle conditions to flourish: moderate temperatures, clean water, protection from radiation, a stable atmosphere. Earth's surface seemed unique precisely because it provided these hospitable conditions. Other worlds, frozen, boiling, bathed in radiation, crushed under immense pressure, seemed obviously lifeless. Then we looked closer at our own planet and discovered we were wrong. Life on Earth doesn't merely tolerate narrow conditions. It thrives in environments that would seem hostile to any form of biology. These organisms, collectively called extremophiles, have rewritten our understanding of where and how life can persist. In the 1980s and 1990s, biologists found that microbial life can survive in extreme environments, such as niches that are acidic, extraordinarily hot, or with irregular air pressure that would be inhospitable to complex organisms. Consider the organisms living near hydrothermal vents on the ocean floor, miles beneath the surface where no sunlight penetrates. In 1977, a team of researchers in a submersible in the Atlantic found arguably one of the most important discoveries of the 20th century: hydrothermal vents, with thriving ecosystems that are not dependent on the sun for energy. These vents spew water heated to over 700 degrees Fahrenheit, laden with toxic chemicals like hydrogen sulfide. The environment is dark, pressurized to hundreds of atmospheres, and chemically hostile. Yet entire ecosystems flourish there. Bacteria, tube worms, shrimp, crabs, all living independently of photosynthesis, deriving energy instead from chemical reactions between seawater and hot rock. The bacteria that form the base of these ecosystems are thermophiles, organisms that not only survive high temperatures but require them to function. A strain of the archaeon Methanopyrus, isolated from a black smoker hydrothermal vent, grows at temperatures of 120 degrees Celsius. At these temperatures, water is near boiling at sea level. Yet these organisms grow, reproduce, and carry out complex metabolism. Their proteins and cellular machinery have evolved to remain stable and functional under conditions that would instantly denature the proteins in our own cells. The discovery of thermophiles revolutionized biology. The discovery of such extremophiles changed scientists' way of looking at life, as the microbes were found in environments where nobody expected life to survive, let alone thrive. If life could exist at those temperatures, what other assumptions about habitability needed to be reconsidered? Temperature extremes work in both directions. Psychrophiles, cold-loving organisms, thrive in permanently frozen environments. Antarctica hosts microbial life in ice, in hypersaline lakes that remain liquid at -25 degrees Celsius, and in rocks a meter beneath the ice surface. Recent research carried out on extremophiles in Japan involved bacteria discovered in Antarctica that feed on air, likely not limited to Antarctica, after discovering the two genes previously linked to their atmospheric chemosynthesis in soil of two other similar cold desert sites. These organisms have adapted their cellular machinery to function at temperatures where chemical reactions normally slow to near stillness. The theoretical limits of life continue to expand as researchers discover new extremophiles. Observed limits for temperature are -20 to 130 degrees Celsius, though the theoretical temperature limit is considered to be between -40 and 150 degrees Celsius. The theoretical limits exist because at -40 degrees, metabolic rates slow so dramatically that cellular processes would take millions of years. At 150 degrees, essential cellular components begin to break down faster than they can be repaired. But temperature is just one variable. Extremophiles challenge nearly every assumption about what life requires. Acidophiles thrive in environments more acidic than battery acid. The microbe Picrophilus can be present in conditions where the pH is as low as 0.06 in solfataric lakes in Japan from where it was isolated. At this pH, the concentration of hydrogen ions is extraordinarily high, conditions that would dissolve most biological molecules. Yet these organisms have evolved protective mechanisms and specialized proteins that function in such acidity. Alkalophiles demonstrate the opposite: adaptation, living in conditions of extreme alkalinity, environments as basic as ammonia. The highest pH maximum of 12.5 was observed for an alkalophilic, aerobic, mesophilic bacterium known as Serpentinomonas species B1. These organisms inhabit environments like soda lakes and serpentinizing systems, where geological processes create extremely basic waters. Radiation resistance provides perhaps the most striking example of extremophile adaptation. Bacteria from Earth, particularly Deinococcus radiodurans, were found to survive for 3 years in outer space, based on studies on the International Space Station. This bacterium can withstand radiation doses thousands of times higher than would kill a human. It accomplishes this through redundant copies of its genome and sophisticated DNA repair mechanisms that rapidly fix radiation damage. The implications for astrobiology are profound. If life on Earth can adapt to such extremes, then the range of potentially habitable environments in the universe expands dramatically. Worlds we once considered too hot, too cold, too acidic, or too irradiated might still harbor life, provided that life evolved different solutions than Earth's surface organisms. Pressure represents another variable where extremophiles have redefined the possible. Barophiles, organisms that thrive under high pressure, live in the deepest ocean trenches, where pressure reaches over 1,000 atmospheres. According to astrophysicist Stein Sigurdsson, there are viable bacterial spores that have been found that are 40 million years old on Earth, and they are very hardened to radiation. These organisms have cellular structures and metabolic processes specifically adapted to function under crushing pressures that would instantly kill surface-dwelling life. Halophiles, salt-loving organisms, live in hypersaline environments like the Dead Sea, where salt concentrations reach saturation. Halophiles require environments with extremely high salt content, such as the Dead Sea, and have developed adaptations to maintain their cellular structure in these hostile settings. These organisms solve the challenge of osmotic pressure through specialized proteins and cellular mechanisms that allow them to maintain function despite the extreme ionic environment. Desiccation resistance pushes survival to another extreme. Some organisms can completely dry out, losing nearly all their cellular water, and remain viable for years. When water becomes available again, they rehydrate and resume normal metabolism. This ability has obvious implications for life on Mars, where liquid water is rare but not absent, or on worlds with seasonal variations that might periodically provide water. The extremophile, with perhaps the most relevance to alien life, is the tardigrade, a microscopic animal known colloquially as a water bear. Tardigrades can survive extreme cold, approaching absolute zero, heat up to 150 degrees Celsius, crushing pressures, the vacuum of space, and intense radiation. There are more than 1,000 species of tardigrades living all over the world and can survive with barely any water. They accomplish this through a process called cryptobiosis, where they essentially shut down all metabolic activity and replace their cellular water with protective molecules. In this state, they can survive for decades, reviving when conditions improve. These capabilities aren't just laboratory curiosities. They demonstrate that life, once established, can adapt to conditions far beyond what we initially imagined possible. Each extremophile represents a solution to a specific challenge: high temperature, low temperature, acidity, alkalinity, radiation, pressure, desiccation. These solutions evolved independently in response to Earth's varied environments. The diversity of extremophiles also tells us something important about the early history of life on Earth. If we consider that the current planetary surface conditions on Earth have only occurred for a short period of time, compared to the existence of life, we might conclude that the extremophilic way of life has actually dominated the evolutionary history of life on our planet. For most of Earth's history, conditions were extreme by current standards: high temperatures, acidic oceans, intense radiation, low oxygen. Life didn't begin in gentle conditions and then adapt to extremes. Life began in extremes, and some lineages later adapted to the relatively moderate conditions of Earth's modern surface. This historical perspective is crucial for astrobiology. It suggests that the extremophile capabilities we observe aren't rare accidents but fundamental features of life that emerge naturally when organisms face environmental challenges. If life arises elsewhere, on Mars, Europa, or planets orbiting distant stars, it will likely develop similar solutions to similar challenges. Researchers have begun to study extremophiles specifically to understand the limits of life. The physical and chemical conditions of Earth's environments exhibit a wide range, much of which, but not all, has been shown to be exploited by microbial life. By mapping where life exists on Earth and where it doesn't, scientists can define the boundaries of habitability with increasing precision. These boundaries then inform the search for life elsewhere. One particularly relevant discovery involves extremophiles living in Mars analog environments on Earth. The Atacama Desert in Chile, one of the driest places on Earth, hosts microbial communities that survive with minimal water. Researchers discovered traces of life in geological samples in the Atacama Desert in Chile that contained hematite and were geologically analogous to Martian soil. Finding life in such Mars-like conditions strongly suggests that if Mars ever had or still has liquid water, even in small amounts, it could support microbial life. Deep subsurface. Environments provide another analog for alien worlds. Microorganisms have been found living in rock formations miles beneath Earth's surface, deriving energy not from sunlight but from chemical reactions between minerals and groundwater. Microorganisms have been found thriving inside rocks up to 1,900 feet below the seafloor, under 8,500 feet of ocean off the coast of the northwestern United States. These organisms exist in complete darkness, isolated from the surface for millions of years, demonstrating that a subsurface biosphere can persist independently of surface conditions. This subsurface life is particularly relevant to moons like Europa and Enceladus, where the surface is frozen but subsurface oceans might exist. Even if the surface is sterile, life could thrive in the dark waters below, just as it does in Earth's deep oceans and subsurface aquifers. The study of extremophiles has also revealed that polyextremophiles, organisms that thrive in multiple extreme conditions simultaneously, are relatively common. Therefore, extremophiles, and in particular polyextremophiles, might be the most abundant life forms on our planet. An organism might be both thermophilic and acidophilic, or halophilic and alkalophilic. These combinations expand the potential habitats for life even further, suggesting that worlds with multiple challenging conditions might still be habitable if organisms can evolve the right suite of adaptations. What's perhaps most calming about extremophile research is the recognition that life, once established, is remarkably robust. It doesn't require pampering. It doesn't need ideal conditions. Given any source of chemical energy, any liquid medium, not necessarily water, though water is common, and any temperature between roughly -40 and 150 degrees Celsius, some form of microbial life could potentially evolve and persist. This doesn't mean life is inevitable in such conditions. It means that if life does arise, the range of environments where it can subsequently spread is far broader than we once thought. The universe offers not just a few perfect worlds, but countless environments where extremophiles, if they exist, could thrive in quiet isolation, unnoticed by any observer looking only for Earth-like conditions. The calm understanding from extremophile research is simple. We've learned to look beyond our assumptions about what life needs. The universe is full of variety, and life, if it's out there, might be thriving in places we haven't yet thought to check, using chemistry we haven't yet imagined, solving challenges we're only beginning to recognize. The search continues, informed by the recognition that life is resilient, adaptable, and more tolerant of extreme conditions than we ever expected. The concept of a habitable zone is deceptively simple. The habitable zone is the orbital region around a star in which an Earth-like planet can possess liquid water on its surface and possibly support life. It's the region around a star where temperatures allow water to exist in liquid form, not frozen solid, not boiled away, but liquid. Since liquid water is essential for all life on Earth, the habitable zone represents the first place astronomers look when considering whether a planet might support life. But the habitable zone isn't a fixed distance. It varies depending on the star's properties. Every star has a habitable zone, but where that zone lies is different for stars of different sizes and brightness. Hot, massive stars emit more energy, pushing their habitable zones outward. Cool, small stars emit less energy, drawing their habitable zones closer in. The zone also shifts over time as stars evolve, becoming brighter and pushing their habitable zones outward over billions of years. In our own solar system, the habitable zone currently extends from roughly 0.9 to 1.5 astronomical units from the Sun, approximately from Venus's orbit to somewhere beyond Mars. At present, the habitable zone of the Sun is estimated to extend from about 0.9 to 1.5 astronomical units. Earth sits comfortably in the middle of this zone, receiving the right amount of solar energy to maintain liquid water across much of its surface. Mars, at 1.5 astronomical units, sits at the outer edge, too cold for widespread liquid water at present, though evidence suggests it once had flowing water when it was younger or during warmer periods. Venus, at 0.7 astronomical units, lies too close to the Sun. The intense solar energy creates a runaway greenhouse effect, with surface temperatures hot enough to melt lead. Yet early in solar system history, Venus might have been habitable, before the greenhouse effect spiraled out of control. This serves as a reminder that being in the habitable zone doesn't guarantee habitability. Other factors matter too. The discovery of exoplanets, planets orbiting stars beyond our Sun, has revolutionized our understanding of habitable zones. Before the first confirmed exoplanet discovery in 1995, we could only theorize about whether other stars had planets. Now we know they do, in abundance. On November 4, 2013, astronomers reported based on Kepler Space Telescope data that there could be as many as 40 billion Earth-sized planets orbiting in the habitable zones of Sun-like stars and red dwarfs in the Milky Way. 40 billion in our galaxy alone. This estimate comes from statistical analysis of the thousands of planets detected by the Kepler Space Telescope, extrapolated across the entire galaxy. Breaking that number down, about 11 billion of these may be orbiting Sun-like stars. The rest orbit red dwarf stars, cooler, smaller, and far more numerous than Sun-like stars. Red dwarfs make up about 70% of all stars in the galaxy, so while their habitable zones lie close to the star, the sheer number of these stars means they contribute significantly to the total count of potentially habitable worlds. These aren't wild speculations. They're based on actual detections and statistical analysis. Using data collected by NASA's Kepler Space Telescope and the WM-Keck Observatory, scientists have estimated that 22% of solar-type stars in the Milky Way galaxy have Earth-sized planets in their habitable zone. More than 1 in 5 Sun-like stars appears to host an Earth-sized planet in the habitable zone. Given that there are billions of Sun-like stars in the galaxy, the number of potentially habitable worlds becomes staggering. Several specific exoplanets have captured particular attention from astrobiologists. Proxima Centauri b, orbiting the star nearest to our Sun, lies within its star's habitable zone. Proxima Centauri b, located about 4.2 light-years from Earth in the constellation of Centaurus, is the nearest known exoplanet and is orbiting in the habitable zone of its star. At just over 4 light-years away, this planet is close enough that future space missions might be able to study it in detail, perhaps even detecting signs of an atmosphere or surface features. The TRAPPIST-1 system has proven particularly intriguing. This system, 40 light-years away, contains 7 Earth-sized planets, 3 or 4 of which orbit in the habitable zone. With 7 Earth-sized worlds in the habitable zone, the TRAPPIST-1 exoplanet system has compelled attention since its 2017 discovery. Having multiple potentially habitable worlds in a single system offers astronomers the opportunity to study different planets with similar initial conditions but potentially different outcomes. Recent discoveries continue to add to the catalog. The new planet named HD 20794d has a mass 6 times that of Earth and orbits a star similar to our Sun located just 20 light-years away, with its orbit placing it within the habitable zone. This super-Earth's proximity and habitable zone location make it a prime target for atmospheric characterization with next-generation telescopes. The Habitable Worlds Catalog maintains an updated list of potentially habitable exoplanets. Some habitable exoplanets detected by radial velocity were considered as stellar artifacts by some studies. This ongoing refinement reflects the challenge of confirming exoplanets and determining their true properties. As observations improve, some candidates are confirmed while others are ruled out or reclassified. The catalog highlights an important point. Being in the habitable zone is necessary, but not sufficient for actual habitability. Inclusion on this list does not guarantee habitability, and in particular, planets must also have other characteristics: appropriate mass, an atmosphere, magnetic field, stable orbit, and more, to truly support life. The unhurried, habitable zone is a starting point, not a guarantee. Different types of stars present different challenges for planetary habitability. Red dwarf stars, while abundant, pose particular concerns. Planets in a red dwarf's comparatively narrow habitable zone, which is very close to the star, are exposed to extreme levels of X-ray and ultraviolet radiation, which can be up to hundreds of thousands of times more intense than what Earth receives from the Sun. This intense radiation could strip away planetary atmospheres over time, potentially sterilizing the surface. However, this doesn't necessarily rule out life on planets around red dwarfs. Atmospheres can be replenished through volcanic activity. Oceans provide shielding from radiation. Subsurface life could exist even if the surface is sterilized. And red dwarfs live for trillions of years, far longer than Sun-like stars, potentially giving evolution plenty of time to find solutions. The characterization of habitable zone planets continues to improve as instruments become more sensitive. The James Webb Space Telescope has already begun studying the atmospheres of planets in habitable zones, detecting molecules that provide clues about surface conditions and potential habitability. Future telescopes will push these capabilities even further, potentially detecting biosignatures, atmospheric gases like oxygen and methane that might indicate the presence of life. One particularly encouraging finding is that planetary systems appear to be the norm, not the exception. The frequency of planets increases to smaller sizes. Terrestrial planets are more common than gas giants. This means that rocky, Earth-sized worlds are actually more common than the giant planets we initially discovered. The early exoplanet detections were biased toward large planets close to their stars because those are easiest to detect. As our methods improve, we're finding that small, rocky planets in habitable zones are abundant. Statistical modeling has refined our understanding of how many habitable worlds might exist. There are currently 20 known exoplanet candidates smaller than 2 Earth radii that fall in their optimistically defined habitable zones. These are worlds that are not only in the habitable zone, but also small enough to be rocky rather than gas giants. They represent the most Earth-like worlds discovered so far, though Earth-like doesn't necessarily mean identical to Earth. The concept of the habitable zone has also evolved to include other considerations beyond temperature. The galactic habitable zone considers where in a galaxy conditions might be most favorable for life. The galactic habitable zone encompasses those regions close enough to a galactic center that stars there are enriched with heavier elements, but not so close that star systems would be frequently disrupted by intense radiation and enormous gravitational forces commonly found at galactic centers. Too close to the galactic center, and supernovae, gamma-ray bursts, and gravitational disruptions would threaten planetary systems. Too far out, and stars lack the heavy elements needed to form rocky planets. Our solar system sits in what appears to be a favorable region of the galaxy, far enough from the center to avoid the most violent events, but close enough that our Sun formed with sufficient heavy elements to create rocky planets. Many other star systems occupy similar positions, suggesting that galactic location doesn't significantly limit the number of potentially habitable worlds. The definition of the habitable zone continues to be refined as our understanding improves. A planet's surface temperature depends not only on its proximity to its star, but also on such factors as its atmospheric greenhouse gases, its reflectivity, and its atmospheric or oceanic circulation. A planet at the outer edge of the habitable zone might maintain liquid water through a strong greenhouse effect. A planet at the inner edge might cool itself through high reflectivity. The habitable zone is more of a guideline than a rigid boundary. Furthermore, life doesn't strictly require surface liquid water. Internal energy sources such as radioactive decay and tidal heating can warm a planet's surface to the melting point of water, and these energy sources can also maintain subsurface reservoirs of liquid water, so a planet could contain life without being within its star's habitable zone. Europa and Enceladus demonstrate this principle. Both are far outside the Sun's traditional habitable zone, yet maintain vast oceans beneath their ice shells through tidal heating from their parent planets. This recognition broadens the search for habitable worlds considerably. Not only do we look at planets in habitable zones, but also at moons of giant planets, even those outside the habitable zone, planets with thick insulating atmospheres, and worlds with active geology that generates internal heat. The sheer abundance of planets in habitable zones creates a sense of statistical inevitability. With tens of billions of potentially habitable worlds in our galaxy alone, and hundreds of billions of galaxies in the observable universe, the total number of places where liquid water and appropriate temperatures coexist reaches into the trillions. Even if only a tiny fraction of those worlds actually develop life, the absolute numbers remain enormous. This abundance doesn't prove that life exists elsewhere, but it removes one of the major potential obstacles: scarcity of suitable environments. The cosmos has provided countless laboratories where the conditions for life might arise. Whether life actually emerges in those laboratories depends on factors we're still working to understand. The probability of abiogenesis, the stability of planetary environments, the role of moons and magnetic fields, and countless other variables. What we do know with increasing certainty is that suitable places are common. The universe has created trillions of worlds where temperatures allow liquid water to exist, where energy from a star bathes the surface, where atmospheres might protect developing organisms, where the chemistry of carbon and water can proceed through its complex pathways. The rest, whether life actually appears, remains an open question. But the stages are set. The theaters are numerous. The cosmic drama of potential life has countless venues where it might play out. Some of the most promising environments for life in our solar system lie not on any planet's surface, but beneath shells of ice, protected from space's harsh conditions in dark oceans that have existed for billions of years. Jupiter's moon Europa has long fascinated astrobiologists. Saturn's moon Enceladus has a subsurface ocean with key ingredients for life: liquid water, carbon compounds, and energy. Though smaller than Earth's moon, Europa appears to contain a global ocean of liquid water beneath its icy crust, an ocean that might contain more than twice the volume of water in all of Earth's oceans combined. Both Europa and Enceladus have subsurface oceans that could harbor life if they have other necessities such as an energy supply, as well as elements and compounds used in biological molecules. The evidence for Europa's ocean comes from multiple sources. The moon's surface shows features consistent with a floating ice shell, cracks, ridges, and chaotic terrain, suggesting that the ice moves and fractures over time. Magnetic field measurements indicate a conducting layer beneath the surface, consistent with a salty ocean. And Europa's slight wobble as it orbits Jupiter can be explained by an ocean decoupling the surface ice from the rocky interior. The ocean exists because of tidal heating. As Europa orbits Jupiter, the massive planet's gravity flexes the Moon, generating friction and heat in its interior. This heat prevents the ocean from freezing solid, maintaining liquid water, even though Europa lies far from the Sun in the outer solar system where temperatures plunge below -160 degrees Celsius. Saturn's moon Enceladus provides even more dramatic evidence of subsurface water. From 2014 to 2017, Cassini investigated Saturn, its rings and moons, and found that Enceladus, only 313 miles in diameter, harbors a liquid water ocean beneath its icy crust that spans the entire Moon. Gazers at its south pole shoot water vapor and ice particles into space, creating one of Saturn's rings. These plumes provide direct samples of the subsurface ocean without need for drilling through the ice. Analysis of the plume material has revealed tantalizing chemistry. Cassini detected new complex organic molecules spewing from Saturn's moon Enceladus, including aliphatic, heterocyclic ester alkenes, ethers ethyl, and tentatively nitrogen and oxygen-bearing compounds. These aren't simple molecules. They're complex, organic compounds that on Earth participate in biochemical pathways. Their presence suggests active chemistry occurring in Enceladus's ocean. Even more intriguing, the plumes contain molecular hydrogen. Cassini's analysis of Enceladus's plumes revealed the presence of hydrogen, carbon dioxide, methane, and complex organic molecules, with hydrogen being especially important because it can serve as an energy source for microbes, much like those found near Earth's hydrothermal vents. This hydrogen likely comes from reactions between hot water and rock at the ocean floor, the same type of reactions that power life at hydrothermal vents on Earth. The presence of hydrogen indicates that chemical energy is available in Enceladus's ocean, independent of sunlight. Phosphorus, essential for life, has also been detected in Enceladus's plume ice grains. Scientists found phosphate, essential for life, in ice grains from Enceladus's ocean. With water, energy, carbon compounds, and phosphorus all present, Enceladus appears to have all the basic requirements for life as we know it. The environments in these subsurface oceans might closely resemble Earth's early oceans. Jupiter's moon, Europa, and Saturn's moon, Enceladus, are currently the most promising candidates among the icy moons as they appear to fulfill the basic requirements for hosting life. The heat that is generated by the tidal pull of the parent planet maintains a subglacial ocean in the liquid state and in direct contact with the rocky core. This direct contact between water and rock enables the water-rock reactions that produce hydrogen and other chemicals that life can use as energy sources. On Earth, entire ecosystems exist at hydrothermal vents on the ocean floor, living in complete darkness, deriving all their energy from chemical reactions rather than photosynthesis. For a long time, sunlight was assumed to be essential for life, but that assumption changed with the discovery of ecosystems on Earth's ocean floor, where life flourishes around hydrothermal vents in complete darkness. These ecosystems demonstrate that sunlight isn't necessary for life. Chemical energy suffices. If similar vents exist on the ocean floors of Europa or Enceladus, similar ecosystems might exist there. The thickness of the ice shell on these moons varies. On Enceladus, the ice might be only a few kilometers thick in some regions, particularly near the South Pole where the geysers erupt. On Europa, the ice shell is estimated to be 15 to 25 kilometers thick. Material on the surface and up to a meter close to it will be degraded by the intense radiation around the parent planet of the icy moon, especially for Europa's parent planet Jupiter. This radiation sterilizes the surface but doesn't penetrate to the ocean below. Remarkably, some biosignatures from these oceans might be detectable even on the surface. Based on our experiments, the safe sampling depth for amino acids on Europa is almost 8 inches at high latitudes of the trailing hemisphere in the area where the surface hasn't been disturbed much by meteorite impacts. Material from the ocean reaches the surface through various processes: plumes, ice convection, fracturing. If that material contains amino acids or other organic compounds, they might survive long enough in the near-surface ice to be detected by future missions. Several missions are planned to explore these ocean worlds more thoroughly. NASA's Europa Clipper mission, launching in the coming years, will make multiple close flybys of Europa, studying its ice shell, ocean, composition, and geology. The spacecraft carries instruments specifically designed to detect organic molecules and assess the moon's habitability. Scientists will place ocean world exploration at the highest priority in NASA's next decade of missions. The European Space Agency's JUICE mission, Jupiter Icy Moons Explorer, will study Jupiter's large moons, including Europa, Ganymede, and Callisto, all of which might harbor subsurface oceans. Ganymede, the largest moon in the solar system, shows evidence of a subsurface ocean that might be sandwiched between layers of ice at different pressures, creating a more complex structure than Europa or Enceladus. Titan, Saturn's largest moon, presents a different type of ocean world. While it has a subsurface water ocean like Europa and Enceladus, Titan's surface features lakes and seas of liquid methane and ethane, a completely different chemistry than water. Titan provides the opportunity to study whether life could emerge in liquid hydrocarbons rather than water, expanding our understanding of possible biochemistries. The prevalence of ocean worlds in our solar system suggests they might be common throughout the universe. If 4 or more moons in our own system harbor subsurface oceans, then moons of giant planets throughout the galaxy might similarly contain hidden oceans. Given that giant planets appear to be common around other stars, the number of ocean worlds in the galaxy could rival or exceed the number of planets in habitable zones. These ocean worlds offer something that surface oceans don't: stability over geological timescales. Earth's surface has undergone dramatic changes over billions of years. Oceans have frozen, continents have moved, atmospheric composition has shifted radically. But beneath kilometers of ice, protected from surface catastrophes, subsurface oceans could remain stable for billions of years, providing consistent environments where life could evolve without repeated disruptions. The ice shells themselves might play a crucial role in habitability. Scientists have speculated that a multi-layered ice crust could develop and serve as a seal or boundary layer between the ocean water and the radiation-blasted surface, creating a more favorable environment for the development and survival of life in the subsurface ocean. The ice protects the ocean from radiation, prevents water loss to space, and might even concentrate organic molecules in certain regions through convection and freezing processes. The timescales involved are staggering. Europa's ocean has likely existed for most of the Moon's 4.5 billion year history. If life emerged in that ocean even 1 billion years ago, it would have had vast stretches of time to evolve and diversify. Evolution on Earth produced complex multicellular life in less time than that. The patience of these hidden oceans is profound. They wait in darkness, potentially harboring life that has been evolving in isolation since the solar system was young. The chemistry occurring in these oceans continues to reveal surprises. The detection of carbon dioxide in Enceladus's plume suggests that carbon cycling occurs in the ocean, reactions that on Earth are often mediated by biology. Whether these reactions are purely chemical or involve biology remains unknown, but the presence of carbon compounds in multiple forms indicates active, complex chemistry. The temperature of these subsurface oceans varies depending on location. Near hydrothermal vents, if they exist, temperatures might reach tens of degrees Celsius, warm enough for Earth-like biochemistry to proceed efficiently. In regions far from heat sources, temperatures might hover just above freezing. This temperature variation creates different ecological niches, much as Earth's oceans support different life forms at different depths and temperatures. Salinity also varies. Europa's ocean is thought to be salty, perhaps similar to Earth's oceans, or even more saline. Enceladus's ocean appears less salty based on plume composition. These differences would influence what types of organisms could evolve, as salinity affects osmotic balance and cellular chemistry. The depth of these oceans is uncertain, but potentially immense. Europa's ocean might be 100 kilometers deep or more, far deeper than Earth's oceans. In such depths, pressure would increase tremendously, creating extreme environments similar to Earth's deepest ocean trenches. If life can adapt to such pressures on Earth, and we know it can, then similar adaptations might occur in Europa's depths. One particularly intriguing possibility is vertical stratification. Earth's oceans show distinct layers with different temperatures, salinity, and chemistry. Europa's ocean might similarly stratify, creating multiple distinct environments within a single ocean. Life might emerge independently at different depths, or organisms might migrate between layers as conditions change. The longevity of these ocean worlds extends into the distant future. Unlike Earth's oceans, which depend on solar heating and the water cycle, subsurface oceans maintain themselves through internal heat and will continue to do so as long as tidal forces persist. Jupiter and Saturn are massive enough that their gravitational influence will continue for billions of years. Europa and Enceladus will likely retain their oceans long after Earth's surface becomes uninhabitable as our Sun evolves. This temporal stability has profound implications. If life exists in these oceans, it might outlast surface life on Earth. These hidden biospheres could become refuges for life in the solar system, persisting through epochs when the Sun's evolution makes the inner solar system inhospitable. In the very long term, subsurface ocean worlds might represent the most stable environments for life in any solar system. The search for life in these oceans will require patience and sophisticated technology. Drilling through kilometers of ice presents enormous engineering challenges. Contamination concerns require extreme care. We must avoid accidentally introducing Earth microbes into pristine alien oceans. But the scientific payoff would be extraordinary. Finding independent life in Europa's or Enceladus's ocean would demonstrate that life arises wherever conditions permit, that Earth is not unique, that the universe harbors biology in multiple places even within our own small stellar neighborhood. For now, these oceans remain mysterious. Hidden beneath ice, possibly teeming with life, possibly sterile, they represent some of the most intriguing targets in the search for alien life. The geysers of Enceladus continue to spray samples into space. Europa's fractured surface continues to shift and crack. And beneath those surfaces, in darkness that has persisted for billions of years, oceans lap against rocky shores, waiting patiently for the day when we might finally discover what they contain. The search for alien life is not a future aspiration. It's happening now, through multiple approaches, using increasingly sophisticated instruments and methods. Astronomers detect exoplanets through several techniques. The transit method watches for the slight dimming of a star when a planet passes in front of it. The radial velocity method detects the wobble planet induces in its star through gravitational tugging. Direct imaging captures actual photographs of planets, though this remains challenging for small, dim worlds. Gravitational microlensing uses the gravity of planets to briefly magnify background starlight. Each method provides different information. Transit observations reveal a planet's size and orbital period. They also allow measurement of the planet's atmosphere. As starlight passes through the atmosphere during transit, molecules in that atmosphere absorb light at specific wavelengths, creating a spectral signature that indicates which gases are present. This atmospheric characterization represents one of the most promising methods for detecting life. Certain combinations of atmospheric gases would be difficult to explain without biology. Oxygen, for instance, is highly reactive and would disappear from Earth's atmosphere within a few million years if not continuously replenished by photosynthesis. Finding substantial oxygen in an exoplanet's atmosphere would suggest biological activity. The James Webb Space Telescope has begun this work in earnest. Since its launch, JWST has studied numerous exoplanets, particularly those that transit their stars and thus allow atmospheric analysis. The James Webb Space Telescope's first detection of carbon dioxide in the atmosphere of a planet outside the solar system was heralded as an important milestone in exoplanet science. Carbon dioxide itself doesn't indicate life, but its detection demonstrates that JWST can identify specific molecules in distant atmospheres. More recently, JWST has detected more intriguing molecules. The James Webb Space Telescope may have detected a molecule called dimethyl sulfide, or DMS, which on Earth is only produced by life. This detection, if confirmed, would be extraordinary. On Earth, DMS is produced primarily by phytoplankton in the oceans. It's released into the atmosphere where it plays a role in cloud formation. Finding it in an exoplanet's atmosphere would strongly suggest biological activity, though researchers emphasize the need for caution and further verification. The planet in question, known as K218b, is a sub-Neptune mass exoplanet orbiting a cool dwarf star approximately 120 light-years away. One of the exoplanets is K218b, a type of exoplanet called a Hycean exoplanet, believed to have a hydrogen-rich atmosphere and a water ocean surface. If confirmed as a Hycean world, a planet with a hydrogen atmosphere and a liquid water ocean, it would represent a new class of potentially habitable environment quite different from Earth. The detection of DMS remains tentative, requiring further observations to confirm. But even the possibility demonstrates how close we are to potentially detecting biosignatures. We're not talking about science fiction scenarios decades in the future. We're talking about observations being made right now, data being analyzed in real time, discoveries that could reshape our understanding of life's place in the universe. Biosignatures come in several types. Some molecules are produced almost exclusively by life, making them strong biosignatures. Others can be produced both biologically and abiotically, making them weak biosignatures that require careful interpretation. The strongest biosignatures involve combinations of gases that wouldn't coexist in the same atmosphere without biological activity to maintain the disequilibrium. On Earth, oxygen and methane coexist in our atmosphere despite chemically reacting with each other. They coexist because biology continuously produces both. Photosynthetic organisms produce oxygen. Methanogenic bacteria produce methane. Without life, one gas would destroy the other within a geologically short time. Finding both gases simultaneously in an exoplanet's atmosphere would strongly suggest biology. Phosphine represents another potential biosignature. Produced by certain anaerobic bacteria on Earth, phosphine was detected in the atmosphere of Venus in 2020, though this detection remains controversial and requires confirmation. If confirmed, it would suggest that Venus, despite its hellish surface conditions, might harbor airborne microbes in its more temperate cloud layers. The search for biosignatures isn't limited to atmospheric gases. Surface features can also indicate life. Seasonal changes in surface color might indicate vegetation or microbial blooms. Unusual concentrations of certain minerals can result from biological activity. On Earth, the massive banded iron formations found in ancient rocks formed when early photosynthetic bacteria produced oxygen that reacted with dissolved iron, causing it to precipitate. Similar geological signatures might be detectable on exoplanets. The chlorophyll edge, a sharp change in reflectivity at the boundary between visible red light and near-infrared, is caused by vegetation on Earth. Plants absorb strongly invisible light for photosynthesis, but reflect strongly in the near-infrared to avoid overheating. Detecting a similar spectral signature on an exoplanet would suggest photosynthetic life, though other explanations might exist. Technology continues to improve. Next-generation ground-based telescopes, the Extremely Large Telescope, the 30-meter telescope, and the Giant Magellan Telescope will have enormous light-gathering power and adaptive optics that correct for atmospheric distortion. These telescopes will be able to directly image some exoplanets and study their atmospheres in detail. Future space missions are also planned. The European Space Agency's Ariel mission will study the atmospheres of hundreds of exoplanets, building statistical understanding of atmospheric chemistry across different types of worlds. NASA's Habitable Worlds Observatory, currently in early planning stages, is specifically designed to search for biosignatures on potentially habitable exoplanets. The search extends beyond exoplanets. Within our solar system, missions continue to explore potentially habitable environments. The Perseverance rover on Mars is collecting rock samples for eventual return to Earth, samples that might contain fossilized evidence of ancient Martian life. The rover has discovered organic molecules and analyzed rock formations that appear to have formed in water, strengthening the case that Mars once had habitable conditions. Mars missions have revealed that liquid water once flowed abundantly on the surface. Valley networks, lake beds, river deltas, all point to a wetter past. Current evidence suggests that early Mars had a thicker atmosphere and warmer temperatures, possibly maintaining liquid water on its surface for hundreds of millions or even billions of years. That's ample time for life to have emerged. Whether life actually did emerge on Mars remains unknown. Even if it did, it might not have survived as the planet lost its atmosphere and dried out. But life is resilient, and Martian organisms might have retreated underground, surviving in subsurface aquifers where liquid water still exists today. The discovery of seasonal methane releases on Mars has fueled speculation about active biology, though geological processes could also explain the methane. The search also includes listening for technological signals, the effort known as SETI, the search for extraterrestrial intelligence. Radio telescopes scan the sky for artificial signals that might indicate technological civilizations. While no confirmed detections have occurred, the search continues to improve in scope and sensitivity. Recent SETI efforts have expanded beyond traditional radio frequencies. Optical SETI searches for brief laser pulses that an alien civilization might use for communication. Searches scan for unusual infrared signatures that might indicate massive engineering projects like Dyson spheres, hypothetical structures that would capture a significant fraction of a star's energy output. The Breakthrough Listen initiative represents the most comprehensive SETI search to date. Using some of the world's most powerful telescopes, it scans millions of stars across a wide range of frequencies. While it has not yet detected confirmed alien signals, it has catalogued millions of interesting signals for further analysis and has developed new techniques for distinguishing artificial signals from natural radio emissions. The absence of detection doesn't mean much at this early stage. The universe is vast, and we've barely begun to search it. If technological civilizations exist but are rare, say one per galaxy, then the nearest might be millions of light years away, far beyond current detection capabilities. Or they might exist at our own level of technology or below, unable to send signals we could detect across interstellar distances. The search also involves looking for technosignatures in other forms: pollution in an exoplanet's atmosphere. Industrial chemicals like chlorofluorocarbons might indicate technological activity. Artificial lighting on the night side of a planet could be detected through careful photometry. Large-scale modifications to a planet's surface might be visible through direct imaging. All these searches share a common feature: patience. We're looking for phenomena that might be rare, distant, and subtle. Success might come next year or might take centuries. But the searches continue because the question is so fundamental. Are we alone? Even a negative answer, confirming that we've looked extensively and found nothing, would tell us something profound about the rarity of life or intelligence. What's particularly encouraging about the current state of the search is the diversity of approaches. We're not relying on a single method or looking in only one place. We're studying exoplanet atmospheres, exploring ocean worlds in our solar system, analyzing Martian rocks, listening for radio signals, searching for optical pulses, and developing new instruments and techniques. This multi-pronged approach increases the chances that, if life exists in detectable forms, we'll eventually find it. The timeline for potential discovery is uncertain, but potentially short. Many researchers believe we have a reasonable chance of detecting biosignatures in exoplanet atmospheres within the next few decades, if not sooner. The steady improvement in instrumentation, the growing catalog of known exoplanets, and the increasing sophistication of our analysis techniques all point toward a time when we might finally have an answer to one of humanity's oldest questions. There's something deeply calming about knowing the search is underway. We're not sitting idly wondering. We're actively looking, measuring, analyzing. Each new observation adds to our understanding, even if it doesn't immediately reveal life. We're learning about planetary formation, atmospheric chemistry, stellar evolution, knowledge that helps us understand our own place in the cosmos even as we search for others. The instruments work quietly, patiently. Telescopes track across the sky. Spacecraft send data across millions of miles. Scientists analyze spectra and calculate probabilities. The search continues in observatories on mountaintops, in university labs, at mission control centers, in the cold depths of space where our robots explore worlds we cannot yet reach. And somewhere in that vast expanse, perhaps on a world circling a distant star, or in an ocean beneath alien ice, or in forms we haven't yet imagined, life might exist. Unaware of our searching, living its own existence according to rules we don't yet know. The search continues, not with urgency, but with methodical thoroughness, knowing that some questions take time to answer and that patience is required when exploring a universe this vast. In 1961, astronomer Frankie Drake proposed an equation to estimate the number of detectable civilizations in our galaxy. The Drake equation wasn't meant to provide a precise answer. Too many variables remain uncertain. But it offers a framework for thinking systematically about the question of alien life. The equation multiplies several factors: the rate of star formation in the galaxy, the fraction of stars with planets, the number of habitable planets per star system, the fraction of habitable planets where life actually emerges, the fraction of life-bearing planets where intelligence evolves, the fraction of intelligent species that develop technology detectable across interstellar distances, and the length of time such civilizations remain detectable. Let's walk through these factors slowly, considering what we now know. The rate of star formation in the Milky Way is reasonably well established. Approximately 7 new stars form in our galaxy each year. This rate has varied over galactic history. It was higher in the past and will decline in the future, but 7 per year represents a reasonable current average. The fraction of stars with planets is no longer speculation. The fraction of stars with planets is essentially 1. Planets appear to be nearly ubiquitous. The more precise question is how many planets each star has, and observations suggest an average of at least 1 or 2 planets per star. The number of habitable planets per planetary system depends on how we define habitable. If we mean planets in the traditional habitable zone with appropriate size and mass, the number is typically 0 or 1 per system, with some systems containing multiple potentially habitable worlds. If we broaden the definition to include ocean worlds like Europa and Enceladus, the number increases significantly, as giant planets often have multiple large moons. Here, the uncertainties begin to compound. What fraction of habitable planets actually develop life? We have a sample size of 1. Earth. Life appeared on Earth remarkably quickly after the planet became habitable, suggesting that abiogenesis might occur readily when conditions allow. Some scientists estimate the probability might be high, perhaps 10 to 50% or more on suitable worlds. Others argue it might be far lower, with Earth representing an improbable outcome. If we take a conservative estimate, say 1% of habitable planets develop life, and apply it to the tens of billions of habitable planets in our galaxy, we still arrive at hundreds of millions of life-bearing worlds in the Milky Way alone. But most of that life would be microbial. The next factor asks what fraction of life-bearing worlds develop intelligent life. On Earth, intelligence of the human type took nearly 4 billion years to evolve. Single-celled life dominated Earth for roughly 3 billion years before multicellular organisms appeared. Complex life then took another 600 million years to produce. Intelligence. This long timeline suggests that intelligence might be rare even on life-bearing worlds. Many planets might host microbial life without ever developing anything more complex. Others might develop complex life but not intelligence. The fraction of life-bearing worlds that produce intelligence could be very small, perhaps 1 in 1,000 or 1 in 1,000,000. Even with such pessimistic assumptions, the sheer number of life-bearing worlds means intelligence might still emerge many times. If 1 in 1,000,000 life-bearing worlds develops intelligence, and there are hundreds of millions of life-bearing worlds in the galaxy, that still yields hundreds of intelligent species in the Milky Way's history. The next factor, the fraction of intelligent species that develop detectable technology, is equally uncertain. On Earth, technological civilization emerged very recently, representing less than 1/10,000th of 1% of the time life has existed on our planet. Some intelligent species might never develop technology. Cetaceans on Earth are intelligent, but lack the manipulative appendages and fire-using capability that enabled human technology. Other pathways to intelligence might similarly fail to produce detectable civilizations. The final factor, the longevity of detectable civilizations, might be the most important and uncertain of all. Humans have been capable of producing detectable radio signals for barely 100 years. Whether we'll continue producing such signals for another 100 years, 1,000 years, 1,000,000 years, or whether we'll destroy ourselves or transition to forms of communication we don't yet imagine remains unknown. If technological civilizations typically last only a few centuries before destroying themselves or evolving beyond detectability, then at any given moment, very few would exist in the galaxy, even if many have existed over its history. Conversely, if some civilizations persist for millions of years, many could coexist at the same time, and the galaxy might be bustling with intelligence we haven't yet detected. Different assumptions plugged into the Drake equation yield wildly different results. Optimistic assumptions suggest millions of detectable civilizations in the galaxy right now. Pessimistic assumptions suggest we might be alone, or that nearest neighbors might be separated by millions of light years. A modified version of the equation focuses specifically on the probability of life rather than intelligence. This version asks, how many planets in the galaxy currently harbor life of any kind, including microbial life? When we remove the requirement for intelligence and technology, the numbers become much larger. Even conservative estimates suggest billions of life-bearing worlds in the galaxy if abiogenesis occurs readily. Statistical approaches continue to refine our estimates. Researchers use Bayesian analysis to incorporate observational data as it becomes available. Each new discovery of an exoplanet in a habitable zone, each new finding about organic chemistry in space, each new understanding of extremophile capabilities updates our probability estimates. One recent approach involves looking at how quickly life appeared on Earth. Life emerged on Earth potentially as early as 4.1 billion years ago, based on evidence from ancient zircon crystals. Life existed on Earth as far back as 3.8 billion years ago, based on geological evidence. Earth formed approximately 4.54 billion years ago. This means life appeared in less than 700 million years, possibly much less. This rapid appearance has two possible interpretations. Either life forms readily when conditions allow, suggesting high probability of abiogenesis, or Earth got extraordinarily lucky, suggesting low probability. Statistical analysis attempting to distinguish between these possibilities remains inconclusive, but tends to favor the interpretation that abiogenesis is moderately probable rather than extremely rare. The anthropic principle complicates this analysis. We exist to ask the question only because life appeared on our planet. Even if the probability of abiogenesis is 1 in a trillion, we'd still find ourselves on one of the rare planets where it occurred. We can't use our own existence to estimate the probability of life elsewhere. It's a selection effect. However, the speed with which life appeared does tell us something. If abiogenesis were extremely difficult, say, requiring multiple improbable events to occur in sequence, we'd expect it to take a long time even on successful planets. The fact that it appeared quickly suggests the process involves fewer bottlenecks than we might fear. Another statistical approach looks at biodiversity on Earth. The extraordinary diversity of life, millions of species filling every conceivable ecological niche, suggests that evolution readily explores possibility space when given time and opportunity. If life exists elsewhere, it too might diversify extensively, creating rich biospheres rather than single surviving lineages. The Great Filter concept provides another lens for thinking about life's probability. Somewhere between non-living chemistry and galaxy-spanning civilizations, there's a bottleneck, a difficult step that few systems pass. Where this filter lies determines our expectations. If the filter lies in the past, say, the jump from non-life to life, or from prokaryotes to eukaryotes, then microbial life might be common while complex life and intelligence remain rare. If the filter lies ahead, perhaps in avoiding self-destruction with advanced technology, then intelligence might be common but short-lived. Evidence from Earth suggests several potential filter locations. The origin of life itself is one possibility. The evolution of eukaryotic cells, which occurred only once in Earth's history through endosymbiosis, might be another. The development of multicellularity, which arose independently several times, might be easier. The emergence of intelligence is clearly difficult, occurring only once among millions of species over billions of years. But none of these apparent difficulties necessarily doom life elsewhere. Earth's sample size is one. What seems difficult here might be easier elsewhere with different initial conditions or different evolutionary pressures. Or conversely, steps that seemed easy on Earth might be difficult elsewhere. The mathematics of large numbers provides a kind of comfort regardless of uncertainties. The observable universe contains so many galaxies, so many stars, so many planets, that even very low probabilities yield meaningful absolute numbers. If the probability of a planet developing intelligent life is 1 in 10 billion, then the observable universe still contains millions or billions of intelligent species. This doesn't guarantee any are close enough for us to detect or contact. Space is vast, and distance separates even abundant phenomena. But it suggests we're probably not alone in any absolute sense. Somewhere, out in that immensity, other forms of life probably exist, even if we never encounter them. The probabilities also change over cosmic time. Early in the universe's history, conditions were less favorable for life. Heavy elements were rarer, stars were hotter and shorter-lived, galaxies were more chaotic. As billions of years passed, conditions improved. More heavy elements accumulated from successive generations of stars. Calmer regions of galaxies formed. Planetary systems had more stable environments. We live 13.8 billion years after the Big Bang. That's enough time for several generations of stars to have formed, lived, and died, enriching the interstellar medium with the elements life requires. It's enough time for quiet, stable planetary systems to have existed for billions of years. In that sense, we might live in the universe's prime era for life, old enough that suitable conditions are common, young enough that stars still shine abundantly. The math suggests patience. Even if technological civilizations are rare and short-lived, their cumulative impact over cosmic history could be substantial. Billions of such civilizations might have existed in the past, with hundreds or thousands currently existing in our galaxy. The distances between them might be too vast for communication, but they needn't be utterly alone. And for microbial life, the simplest, most robust form, the numbers suggest abundance. If life arises with any reasonable probability on suitable worlds, then the galaxy hosts billions of biospheres. Most would be simple, some might be complex, a few might be intelligent. The cosmic ecosystem would be rich and varied, with life exploring countless pathways across countless worlds, each writing its own chapter in the story of biological existence. The uncertainty in these calculations doesn't undermine their value. They remind us that we're asking questions at the frontier of knowledge, where uncertainty is expected and humility appropriate. The numbers don't provide definitive answers, but they frame the question in ways that help us think clearly about probability, scale, and what we might realistically expect to find as we continue our search. The question of life's origin has always assumed that life begins independently on each world where it exists. But there's another possibility, one that scientists take seriously: panspermia, the hypothesis that life can travel between worlds, seeding planets with biology that originated elsewhere. The idea isn't new. It dates to ancient Greece and was revived in modern times by scientists including Svante Arrhenius and Fred Hoyle. But only recently have we gathered evidence showing that panspermia is physically plausible, not merely speculative. The mechanism is straightforward. When a large asteroid or comet impacts a planet with sufficient force, the explosion can eject material into space. Rock fragments escape the planet's gravity and drift through the solar system. Eventually, some fragments fall onto other worlds, delivering whatever they contain, including potentially microorganisms. We know this happens because we've found Martian meteorites on Earth. Approximately 175 Martian meteorites have been found as of 2021. These rocks were blasted off Mars by impacts, traveled through space for millions of years, and eventually fell to Earth. If rocks can make this journey, so can any microbes they contain. The question becomes whether microbes can survive such a journey. The environment of space is hostile: vacuum, radiation, extreme cold, no nutrients. But experiments have shown that some organisms can endure these conditions, at least for limited periods. Bacterial spores in particular show remarkable durability. Bacteria from Earth, particularly Deinococcus radiodurans, were found to survive for 3 years in outer space based on studies on the International Space Station. 3 years isn't enough to travel between stars, but it's sufficient for transfer between planets in the same solar system. The journey from Mars to Earth typically takes millions of years, longer than we'd expect any microbe to remain viable, even in a protective rock. However, much faster transfers are possible. Computer simulations show that a tiny fraction of material ejected from Mars reaches Earth in less than a year, while larger fractions arrive within tens or hundreds of thousands of years. If a large population of microbes was ejected, even a tiny survival fraction could seed a destination planet. Protection matters enormously. Microbes on the surface of a rock would be sterilized by ultraviolet radiation and cosmic rays. But organisms buried within the rock, shielded by centimeters or meters of stone, would receive far less radiation. The rock itself provides protection, and the cold of space essentially puts organisms into suspended animation, slowing metabolic decay to near zero. Laboratory experiments have subjected bacterial spores to simulated space conditions, vacuum, radiation, temperature extremes, and found survivors even after years of exposure. Some spores remained viable after being subjected to conditions harsher than most of the transfer journey would involve. Tardigrades provide an even more striking example. These microscopic animals can survive the vacuum of space, temperatures near absolute zero, and radiation doses that would kill most other organisms. While tardigrades are too complex to have plausibly transferred between worlds in the past, their abilities demonstrate that life can evolve extraordinary protective mechanisms against space's challenges. If panspermia occurred within our solar system, it has profound implications. Life might have originated only once, perhaps on Mars, perhaps on Earth, and then spread to other worlds through natural processes. This would explain why life appeared so quickly on Earth after the planet became habitable. Perhaps it didn't need to originate here. Perhaps it arrived already formed from somewhere else. Mars is actually a more plausible origin point than Earth for several reasons. Early Mars appears to have become habitable earlier than Earth, while our planet was still suffering through the late heavy bombardment, a period of intense asteroid impacts that might have repeatedly sterilized the surface. Mars, being smaller and cooling faster, could have offered stable habitable conditions first. Life originating there could have later transferred to Earth. If this occurred, we might share a common ancestor with any life that currently exists on Mars. Future missions that discover Martian microbes will analyze their biochemistry carefully. If Martian life uses DNA, the same amino acids, and the same genetic code as Earth life, it would strongly suggest a common origin, either panspermia from Mars to Earth, Earth to Mars, or both planets seeded from some third source. Alternatively, if Martian life uses completely different biochemistry, different genetic polymers, different amino acids, different coding systems, it would demonstrate independent origin. Two independent origins of life within one solar system would strongly suggest that abiogenesis occurs readily, boosting estimates of life's prevalence throughout the universe. Panspermia could work in other directions. Europa or Enceladus might have received terrestrial or Martian microbes through the same impact ejection mechanism. Given the billions of years available and the numerous large impacts that have occurred, material has certainly transferred among most bodies in our solar system. Whether any of that material contained viable organisms remains unknown. The possibility extends beyond our solar system, though the challenges become far greater. Interstellar panspermia would require organisms to survive millions or billions of years in space, endure far higher radiation doses, and survive entry and impact on a destination planet. Some scientists consider this implausible. Others argue that in rare cases, perhaps inside comets or large asteroids that protect their interiors, survival might be possible. If interstellar panspermia occurs, life could spread gradually through the galaxy over billions of years. A successful biosphere on one planet seeds nearby systems. Some of those seeds succeed, creating new biospheres that in turn seed further systems. Over geological time, life could propagate across significant volumes of space, even without intelligent agents deliberately spreading it. This process has been called radiopanspermia when considering that small particles or spores might be pushed by radiation pressure from stars, while larger rocks would be required for successful transfer. The concept illustrates how life might disperse through natural processes without requiring intentional seeding by technological civilizations. Some scientists have proposed directed panspermia, intentional seeding of other worlds by intelligent species. A civilization might send spacecraft carrying microorganisms to nearby star systems, deliberately creating new biospheres. While we have no evidence of this, it represents a possibility that some civilizations might choose to pursue. The implications of panspermia for the search for life are mixed. On one hand, if life spreads between worlds, it might be more common than if each biosphere must originate independently. Abiogenesis might be extremely rare, but if successful instances spread their biology to other worlds, life could still be relatively abundant. On the other hand, all that life might share a common biochemical foundation, reducing the diversity of biological solutions we might discover. Current evidence doesn't prove or disprove panspermia. We know material transfers between planets. We know some organisms can survive space exposure for limited periods. We know life appeared quickly on Earth, consistent with either rapid abiogenesis or external delivery. But we don't yet have definitive evidence of organisms surviving the full journey from one world to another within our solar system, much less between systems. Future Mars sample return missions might provide crucial evidence. If we bring pristine Martian samples to Earth and discover organisms with terrestrial-like biochemistry, panspermia becomes the leading explanation. If we find organisms with alien biochemistry, independent origin seems more likely. If we find no organisms at all, the question remains open. The concept of panspermia adds another layer of possibility to our understanding of life in the universe. Rather than viewing each world as isolated, we can think of solar systems, and perhaps even larger volumes, of space, as potentially connected by the slow exchange of material. Life might be less lonely than we imagine, not just in the sense of arising independently in many places, but in the sense of spreading, sharing, connecting across the vast distances between worlds. This sharing needn't be rapid or efficient. Over millions of years, occasional successful transfers would suffice. The cosmos operates on such immense timescales that even extremely rare events become probable given sufficient time. A 1 in a billion chance of successful transfer between planets becomes likely when billions of impacts occur over billions of years. Whether panspermia occurred in our solar system's past, whether it continues today, whether it operates on interstellar scales, these questions await answers. But the possibility remains open, suggesting that life's story might involve not just isolated origins, but also journeys between worlds, carried silently through space in the hearts of rocks, waiting patiently for the chance to seed new worlds with the ancient chemistry of existence. When we think about the search for alien life, our human perspective can mislead us. We think in human lifespans, decades, or at most, in terms of recorded human history, thousands of years. But life operates on entirely different timescales, and understanding those timescales provides perspective on both the challenges and inevitabilities of the search. Earth's geological history spans 4.5 billion years. That's 4,500 million years. Written out, 4,500,000 years. The number is difficult to grasp intuitively. Let's try to find perspective. If we compressed Earth's entire history into a single 24-hour day, humans would appear in the last minute before midnight. Civilization would exist for less than a second. Our species has been technological for a fleeting instant at the very end of a very long day. Life, on the other hand, appears early in that compressed day. By mid-morning, representing several hundred million years after Earth's formation, life had already emerged. Simple cells dominated for most of the day. Complex multicellular life appeared only in the evening. The entire story of plants, animals, and ecosystems we're familiar with occupies less than the final 2 hours of that metaphorical day. This temporal perspective has profound implications for astrobiology. Life doesn't need billions of years to appear. It appeared quickly on Earth. But life might need billions of years to become complex, and billions more to produce intelligence. The timeline from chemistry to consciousness is measured in eons. Other worlds at different stages of this timeline would show different characteristics. A planet where life appeared 1 billion years ago might still be entirely microbial. Its biosphere, simpler than Earth's, was 3 billion years ago. A planet where life appeared 6 billion years ago, if such planets exist around older stars, might host organisms whose evolution has proceeded 2 billion years beyond Earth's current state. What would 2 billion years of additional evolution produce? We can't know. Earth's life 2 billion years ago was microbial. In the 2 billion years since, it progressed through the evolution of eukaryotes, multicellularity, plants, animals, fungi, and ultimately intelligence. 2 billion more years of evolution from today's starting point might produce outcomes we can't imagine. The pace of evolution varies enormously. Sometimes evolution proceeds slowly. Morphologies remain stable for tens or hundreds of millions of years. Other times, evolution accelerates. The Cambrian explosion saw most major animal body plans appear within 10 to 20 million years, a blink of an eye in geological time. Environmental changes, mass extinctions, new ecological opportunities, all influence evolutionary pace. This variable pace means we can't assume that all life-bearing planets follow similar trajectories on similar timescales. Some might evolve complexity quickly, others might remain simple for billions of years before suddenly diversifying. Some might develop intelligence early, others never. The pathways are contingent, dependent on countless factors we're only beginning to understand. Geological stability matters too. Earth has benefited from unusual stability. Our large moon stabilizes our axial tilt, preventing wild climate swings. Our magnetic field protects the atmosphere from being stripped away by solar wind. Our position in the solar system, just the right distance from Jupiter to benefit from its protecting influence against comets while not experiencing too many gravitational disruptions, has favored long-term stability. Worlds lacking these stabilizing factors might experience frequent catastrophes that repeatedly reset the evolutionary clock. Life would persist. Extremophiles would survive in protected niches, but complex ecosystems might never establish, repeatedly destroyed before they could diversify. Conversely, worlds with greater stability than Earth might see even more consistent evolution. A planet with less geological activity might avoid mass extinctions, allowing evolutionary lineages to explore possibility space for billions of years without major disruptions. The result might be biospheres of stunning complexity, with ecological relationships refined by unimaginable spans of time. The timeline also favors life in another sense: patience in the search. We've been seriously searching for alien life for only a few decades. We've had the technology to detect exoplanets for barely 30 years. We're in the earliest days of atmospheric characterization and biosignature detection. In terms of the cosmic timeline, we've barely begun. Scientists often speak of the cosmic calendar, the history of the universe compressed into 1 year. In this calendar, the Big Bang occurs at midnight on January 1st. Our solar system doesn't form until September 1st. Life on Earth appears by late September. Dinosaurs appear in mid-December. Humans appear at 11:58 p.m. on December 31st. All of recorded human history occupies the final seconds before midnight. In this context, expecting immediate success in our search for alien life seems premature. We're conducting our search in a tiny sliver of time. If we don't find life this decade or next, it means little. The search might require centuries or millennia to yield results, and that's fine. The universe isn't going anywhere. The age of the universe itself, 13.8 billion years, provides ample time for life to have emerged many times in many places. Some of that life might be billions of years old, persisting in forms that have adapted to every conceivable niche on their home worlds. Other life might be young, newly emerged on planets orbiting young stars, just beginning its evolutionary journey. This temporal spread means that if we ever discover multiple instances of life, they'll almost certainly be at vastly different stages of development. The probability of finding another civilization at our exact level of advancement is essentially zero. We'd find either much simpler life, microbes or simple multicellular organisms, or much more advanced civilizations that have been technological for millions of years. The timeline of star formation also matters. Stars have been forming throughout cosmic history, but the rate has declined over time. The universe produced stars most prolifically several billion years ago. Those stars now host mature planetary systems where life, if it emerged, has had billions of years to evolve. The stars forming today are latecomers, their planets young and still settling into stable configurations. This suggests that we might live in an era where life-bearing worlds are reaching peak maturity. The first generations of truly stable, habitable planets formed billions of years ago and now potentially host advanced biospheres. Later generations of habitable planets are still developing. We exist in a middle era. Our sun is neither among the oldest nor the youngest, which might be typical of life-bearing systems. The future timeline stretches even more incredibly than the past. Our sun will continue shining for approximately 5 billion more years before evolving into a red giant. Other stars will shine longer. Red dwarfs might last 10 trillion years or more. Life that emerges on planets around red dwarfs could have almost unimaginable spans of time to evolve. In such vast futures, assuming life persists and spreads, what might emerge? Evolution would explore every possible biological solution. Ecosystems might achieve levels of complexity we can't conceive. Intelligence might appear many times in many forms. The cosmos has time, vast, patient time, for every possibility to eventually occur somewhere. This perspective provides a kind of serenity. We don't need to find alien life immediately. The universe isn't running out. If life is out there, and the evidence increasingly suggests it probably is, then it will continue existing whether we discover it next year or in 1,000 years. Our timeline of discovery is irrelevant to life's actual existence. The search continues at its own pace, driven by improving technology and growing understanding. Each decade brings better instruments, more sensitive detection methods, deeper insights into where and how to search. The probability of discovery increases with time, not just because we're looking longer, but because we're looking smarter, guided by accumulating knowledge about planets, stars, chemistry, and life itself. Some searches might take generations. The study of exoplanet atmospheres requires decades of observations to build up sufficient signal. Sample return missions from Mars or Europa might not. Launch for years and take more years to return. SETI searches scan billions of star systems, a project that could continue for centuries before achieving comprehensive coverage. But that's acceptable. The timeline favors patience. We're not racing against a clock. We're gradually, methodically expanding our knowledge, confident that if we continue improving our capabilities and broadening our search, eventually, perhaps soon, perhaps distant, we'll find what we're looking for, or it will find us. And even if we never achieve direct contact with alien life, the search itself teaches us profound lessons. It reveals the universe's structure and history. It clarifies life's requirements and possibilities. It places our own existence in cosmic context. The journey of searching matters as much as any destination we might reach. The timeline reminds us that we're part of a much larger story. Life emerged on Earth 4 billion years ago. It persisted through countless challenges. It evolved into forms of staggering diversity. It eventually produced beings capable of wondering about life elsewhere. And now, in these brief decades of early technological maturity, we're beginning to develop the means to answer that question. Other worlds might be following similar timelines, or completely different ones. Some might be behind us, their biospheres still simple, their surfaces devoid of anything we'd recognize as complex life. Others might be far ahead, their evolution having explored pathways Earth's life has yet to discover or never will. The vast span of cosmic time accommodates all these possibilities. It allows for every variation, every pathway, every outcome. Life, wherever it exists, unfolds according to its own schedule, shaped by the unique conditions of its world. The timeline favors life not because it makes life inevitable at any moment, but because it provides such immense spans for the unlikely to become probable, for the possible to become actual, for the potential to be realized. We exist in our small moment of that timeline, looking outward, looking inward, seeking to understand our place in existence. The timeline stretches behind us into deep history and ahead into unimaginable futures. And somewhere in that immense span, other moments of awareness might exist. Other beings looking up at their own skies, asking their own questions, conducting their own searches. The timeline favors life. It favors diversity. It favors the slow, patient unfolding of possibility into reality, and it favors those who search with patience, knowing that some questions require time to answer, and that the answer, when it comes, will be worth the wait. We've journeyed through vast spaces and deep time, through statistics and chemistry, through ice-covered oceans and distant atmospheres. What emerges from all these facts, these measurements, observations, discoveries, isn't drama or urgency, but something quieter and perhaps more profound. The universe creates life-friendly conditions regularly. Not everywhere, not always, but regularly enough that the improbable becomes plausible, that the isolated becomes less lonely, that the question of whether we're alone receives an answer weighted toward no, even if we haven't yet confirmed it. The ingredients exist in abundance, organic molecules drifting through space, delivered to worlds by asteroids and comets. The environments exist in profusion. Billions of planets in habitable zones, ocean worlds beneath ice, possibly niches we haven't yet imagined. The time exists in immensity. Billions of years for chemistry to explore its possibilities, for evolution to solve its challenges, for complexity to emerge from simplicity. Life on Earth thrives in extremes we once thought impossible. It adapts, persists, survives. This resilience suggests that life elsewhere, if it exists, might similarly persist in environments we haven't thought to check, using chemistries we haven't fully considered. The search continues patiently. Telescopes scan exoplanet atmospheres. Rovers explore Martian rocks. Spacecraft fly to ocean moons. Radio telescopes listen to the cosmos. Scientists analyze data, refine models, prepare new missions. Each effort adds another piece to the puzzle, even when it doesn't immediately reveal the answer we seek. What's certain is that we live in a universe far more abundant, more varied, and more potentially alive than previous generations imagined. The cosmos isn't hostile to life. It's simply vast. So vast that even abundant phenomena can seem rare. So vast that distance separates even common occurrences. Whether we're alone remains uncertain. Whether we'll discover alien life in our lifetimes remains unknown. But the evidence suggests we live in a universe where life, if not inevitable, is at least plausible. Where the building blocks assemble naturally. Where suitable environments exist in great numbers, where time allows for the gradual emergence of complexity. The search will continue, perhaps for decades, perhaps for centuries, and one day, maybe soon or maybe in a future we can't yet imagine, we might find an answer. A biosignature in an exoplanet spectrum, microbes in Martian soil, complex molecules in Europa's ocean, a signal from a distant star. Or perhaps none of these things, but instead a gradual accumulation of evidence that life, while not universal, occurs with enough frequency that we can reasonably expect it exists somewhere out in that tremendous expanse. For now, we continue our observations, we refine our instruments, we expand our search, and we do so with the recognition that we're asking one of the most profound questions our species has ever posed, and that profound questions sometimes require patience and time to answer fully. The universe waits, as it has waited for billions of years, operating according to its own laws and timescales. Life, wherever it exists, follows those same laws, chemistry becoming biology, simplicity becoming complexity, individual forms becoming ecosystems. The processes are universal. The outcomes might be too. And so we watch and listen and explore, not with anxiety, but with careful attention, not with urgency, but with methodical thoroughness. We search because we can, because the question matters, because understanding our place in existence requires knowing whether that existence is shared. The cosmos, vast and ancient, continues its slow evolution. Stars shine, planets orbit, chemistry proceeds, and somewhere in all that immensity, perhaps on worlds we've observed, or perhaps on worlds we have yet to discover, life might exist, quietly, patiently, following its own pathways through time. That possibility, grounded in evidence and measurement rather than wishful thinking, provides a kind of comfort. We're probably not alone. We might discover that truth in our lifetimes, or it might take longer. But the question has an answer, and we're working toward finding it, guided by science, driven by curiosity, supported by the recognition that the universe seems configured in ways that allow life to emerge when conditions permit. The search continues, the cosmos endures, and somewhere in the intersection of those two facts, an answer waits. Thank you for spending this time exploring these ideas. If you found this journey through the possibilities of alien life calming and thought-provoking, I'd be grateful if you'd like the video and consider subscribing to Nebula Nights. There's so much more to explore together. The quiet mysteries of the cosmos, the gentle facts that inspire wonder without requiring urgency. Until next time, rest well and remember, the universe is vast, patient, and probably not as empty as the night sky sometimes makes it seem.